So hi everyone, thank you for joining our tutorial this Tuesday. So this will be a tutorial for early year students on respiratory physiology by Lisa Dwyer-Joyce. So just some house rules for everyone tuning in. If you are on the Zoom chat, there are questions, so polls will pop up in the app, so you don't have to worry about going somewhere else. In regards to asking questions or comments, if you have anything referring to what is happening right now, please put it in the chat. If you have any questions that can wait to the end or just more general questions, put them in the Q&A function. And at the end of our session, all of our tutorials and slides are uploaded to the Becoming a Doctor website. So make sure you're signed up to that and you can view it again later at your own pleasure. So now I'll just hand it over to Lisa and good luck, everyone.
Okay, thank you. I'll just start sharing my slides. Okay, so my name's Lisa, like I was just introduced, and I'm a third year medical student at Bristol University. And I'm just going to do a respiratory physiology session for you guys today, and it's aimed at preclinical years, or those of you whose exams are more focused on how the body functions than to do with diseases. So yeah, this is a tutorial for going over some of that stuff. Just a quick disclaimer there. So the objectives that I'm hoping to cover in today's session are to understand more about how gases are exchanged in the lung, and more about VQ ratios and how that affects gas exchange, to understand how carbon dioxide and oxygen are transported in the blood, and to understand the mechanics of ventilation and how ventilation is controlled. So this is a revision session, so hopefully these are topics that you'll have already covered in your teaching, but it's just something to kind of go over that and just consolidate your learning and make sure it's just nice and fresh in your minds before exams coming up. And yes, if anyone has any questions, like we've already said, just put them in the chat. So I'm going to start with gas exchange. So gas exchange takes place in the lungs where the alveoli are, which are the little sacs of air. And it takes place between those alveoli air sacs and the surrounding pulmonary capillaries. And gas exchange mostly takes place by simple diffusion, which is down a concentration gradient or a partial pressure gradient. So that's from an area of high partial pressure to an area of low partial pressure. And when we're talking about gas exchange in the lung, there are two main gases. So oxygen moves from the external environment through the conducting airways into alveoli. And then because there are high partial pressures of oxygen in the alveoli and there is a low partial pressure in the surrounding blood in the capillaries, oxygen will then move from the external environment from the alveoli into the blood down the partial pressure gradient. But carbon dioxide is going the other way because it's being produced at the tissues during respiration. So carbon dioxide gets produced at the tissues and it gets loaded onto the blood and taken to the pulmonary capillaries, where it then goes from the higher partial pressure in the pulmonary capillaries into the lower partial pressure of carbon dioxide into the alveoli. So you've got the two gases moving in opposite directions here. You've got oxygen moving from the alveoli into the blood and carbon dioxide moving from the blood and into the alveoli. And the rate of gas exchange depends on different things. So it depends on exchange surface thickness. So the thinner the exchange surface barrier, the faster the rate of the diffusion. Because if the gases have to travel a shorter distance, it's going to take less time. So in the lung, that is maximized by having a very thin capillary, alveolar capillary membrane, which means that the alveolar epithelium and the capillary endothelium are very thin. So the gases do not have to travel very far at all. And that just means the gas exchange rate is as quick as it can be. Gas exchange rate also depends on exchange surface area. So the larger the surface area, the faster the gas exchange, just because there's more area for the gas to travel across. So in the lungs, this is maximized by having lots and lots of small alveoli that together form a very large surface area for the exchange. And that's just how it's speeded up in the lung. And exchange rate also depends on concentration gradient. So that's the difference between the concentration on either side of the membrane. And the steeper the concentration gradient, the faster the gas exchange rate. So in the lung, this is maximized by making sure that your lungs are constantly ventilated. So it's taking away carbon dioxide and bringing new oxygen. And blood is constantly moving through the pulmonary capillaries. And that just means that you have constant renewal of that concentration gradient so gases can keep exchanging. So the lungs are adapted for very efficient gas exchange because that's what they're for. And things like different diseases can affect surface thickness and surface exchange area and slow gas exchange rate. So now I'm going to move on to ventilation and perfusion. So the ventilation rate is the volume of gas that is delivered to the alveoli in a given time. And you would calculate this normally by working out the tidal volume, which is just the volume of air inhaled or exhaled in normal breath. And you would minus dead space from this. Now, dead space is the volume of gas in the airways that isn't involved in gas exchange. So that'd be things like airways, air in your trachea and your bronchi that aren't in the alveoli. So you work out the amount of gas that's actually involved in gas exchange, and then you would times that by the respiratory rate. And then you have your ventilation, which is given the symbol V. And perfusion is the total volume of blood reaching the pulmonary capillaries in a given time period. So together you can make a VQ ratio, which is the volume of gas delivered to the alveoli over the total volume of blood reaching the pulmonary capillaries. So a low VQ ratio would mean that there would be more blood flow in the capillaries than there would be ventilation in the alveoli. And a high ratio would mean that there is less blood flow than there is ventilation. The ideal ratio would be 1, and that would mean that you have exactly the same amount of ventilation as you do perfusion. And that is just the most maximum gas exchange that you'll get if you just have this perfect 1 VQ ratio. Sadly, in the lungs, you don't have this perfect one VQ ratio everywhere. In fact, the VQ ratio actually varies depending on what part of the lung you're in. So the VQ ratio is lowest at the bottom of the lungs, as you can see in this little diagram. And that's because there is more perfusion than there is ventilation at the bottom of the lungs. And then as you travel up to the apices of the lungs, so the top of the lungs, your VQ ratio increases because you're getting more ventilation than you do perfusion. And that's just to do with the effects of gravity on the lung and the fact you've got decreasing perfusion as you travel up towards the top of the lungs. But the average VQ value for a human lung, just the total lung in total, would be 0.8. So it's not quite 1, but it's quite close. So you'd still get enough gas exchange taking place. And that's just in an average human lung. So if you've got differences between ventilation and perfusion, you get changes in the VQ ratio, and these are called VQ mismatches. And they would decrease the amount of gas exchange that you have taking place. So certain diseases can cause VQ mismatches. A low VQ ratio would mean that there would be more blood flow in the capillaries than ventilation to the alveoli. So any kind of diseases that would cause less ventilation to take place would cause a low VQ ratio. And that could be things like airway obstruction or asthma, COPD, anything where you've got less ventilation. And if you look at this little second image here that says airway obstruction, you can see that you've got something that's blocking your alveoli, so there is less ventilation to this, there's no ventilation to this alveolus, but you've still got blood flowing through the capillary. And that means you've got a VQ ratio of 0. And what that creates is something called a shunt. And that's basically when you've got no ventilation in the alveolus, but you've still got perfusion to the capillaries. But a high VQ ratio would mean that there is less blood flow than there is ventilation. So that would be caused by any kind of diseases that would reduce the blood flow. So that would be things like a pulmonary embolus, which would then block your capillaries, or it could be things like low blood pressure, reducing your perfusion there. So if you look at the third picture here, which is your pulmonary embolus, you can see you've got something that's completely blocking your capillary. So there's no perfusion, but you've still got ventilation of this alveolus. And that gives you an infinite VQ ratio. And what you've created here is dead space in this alveolus because the gas that is entering that alveolus is no longer taking place in gas exchange because you've got no blood flow there. to receive the gases. So VQ mismatches are important because if you have one, it can cause a reduction in the amount of oxygen that you're getting in the body, causing hypoxia. And it can also cause an increase in carbon dioxide in the blood. So VQ mismatches is something to watch out for when you're dealing with people that might have pulmonary diseases. I've got a practice question here for everyone, just about that section so far. So I'm just going to check, I've got a question here in the Zoom chat. I thought that a low VQ caused PE and a high VQ COPD. I'm going to have to check that one for you. I'm not too sure. But I'll double check because that might be correct. So sorry about that. So here's your practice question. So this is your 25-year-old male that has a respiratory rate of 15 breaths per minute and a tidal volume of 500 milliliters. His physiological dead space is 150 milliliters and the perfusion rate to his lungs is 6 liters per minute. What is his VQ ratio? So you might need to get a calculator for this one, guys, unless you're very good at doing mental maths. And I'll just open the polling. Okay. I'll give you guys a little bit of time to answer this one just because it involves some calculations. Okay, I'll just give you 10 more seconds here. Okay, so well done, guys. Most of you got this one right. I'll just share the results. So as you can see, the right answer here is 0.875, your VQ ratio. And I'll just show you that calculation for you here. So you would put in your tidal volume, which is 500 milliliters, minus your dead space. That gives you 350 milliliters. I'll convert that into liters and then times that by the respiratory rate. Gives you 5.25, and you're given the perfusion, which is 6. And that gives you a ratio of 0.875. Okay, so gas transport in the blood. So oxygen can be transported in the blood in two main ways. The majority of oxygen is bound to hemoglobin, forming oxyhemoglobin. So that's where the majority of oxygen is transported. But a small amount of it is dissolved in the plasma as well. So if you want to work out the total oxygen content of the blood, you would add together the amount that is bound to hemoglobin and the amount that is dissolved in the plasma. You can go one step further than this and work out the oxygen delivery. And that would be multiplying the oxygen content of your blood times by the cardiac output. And that's just how you work out the amount of oxygen that's being delivered to your tissues. And this is your hemoglobin molecule. So hemoglobin is a protein and it's a tetramer, so it's formed of 4 polymer chains, 2 beta chains and 2 alpha chains. And each chain forms a subunit, and each subunit has a heme group. And the heme group contains an Fe2 plus ion, which can bind to an oxygen molecule. So because there are 4 subunits and 4 heme groups, you then get the hemoglobin's capacity to carry 4 oxygen molecules. Now, hemoglobin's affinity to bind oxygen is ability to bind oxygen, is known as its affinity. And the higher affinity, the easier it is for oxygen to bind to haemoglobin. Now, the affinity of haemoglobin for oxygen can actually vary based on the concentrations of oxygen or the partial pressure of oxygen in the surrounding gas. And this is because of something called cooperative binding that oxygen and haemoglobin undergoes. And this is because once the first oxygen molecule has bound to that first subunit, it changes the shape of the haemoglobin slightly and makes it easier for subsequent oxygen molecules to bind. This is called cooperative binding. And if you plot a graph of haemoglobin saturation over partial pressure of oxygen, then you get this sigmoid curve here called the oxygen dissociation curve. And as you can see, at lower partial pressures of oxygen, you get a lower saturation of haemoglobin. But at higher partial pressures, for example, in the lungs, you get a much higher saturation of haemoglobin for oxygen. And that just means at areas of high oxygen, like in the lungs, you get as much haemoglobin saturation as possible. And then in areas of low partial pressure of oxygen, for example, at the tissues, you get a lower saturation of oxygen and more is dissociated from the hemoglobin and into the tissues to get used. So different factors can actually change hemoglobin's affinity for oxygen. So if there is a decreased affinity for oxygen, you get a right shift in the curve, which would look like this little red line here. And that's if you have a decreased affinity for oxygen for hemoglobin. If you have increased affinity for oxygen, you get a left shift in the curve, or this blue line here. So as you can see, for any given partial pressure of oxygen, let's say you're at 20 here, if there's a decreased affinity for in hemoglobin for oxygen, you get a lower level of oxygen saturation. If there's a decreased affinity, and if you've got an increased affinity, which would be the blue line, you get a much higher oxygen saturation for hemoglobin. And the factors that can affect hemoglobin's affinity for oxygen is pH, or concentration of H+ ions. So the higher the concentration of H+ ions, and that would be a lower pH, the lower the affinity of oxygen of hemoglobin for oxygen is. So it causes a decrease in affinity. And this decrease in affinity that corresponds to an increase in H+ concentration is called the Bohr shift. And what that means is, in the tissues where you're producing carbon dioxide, you're getting a decrease in pH because carbon dioxide is acidic, and hemoglobin is getting a decreased affinity for oxygen. And that causes more oxygen to dissociate to then be used up by the tissues. And you would get the reverse as well if you had a decrease in carbon dioxide concentration and therefore H+ ions. And you're getting an increase in pH, and that will cause an increase in affinity. You also get something called 2,3-diphosphoglycerate, which can affect the affinity for oxygen. So 2,3-diphosphoglycerate is a product of glucose metabolism. And if you've got increasing concentrations of it, it causes a decrease in affinity, so it will cause that right shift. And 2,3-diphosphoglycerate is produced when the body is at high altitudes and when you require more oxygen because there's lower partial pressure, atmospheric partial pressures of oxygen. And so you get a decreased affinity of hemoglobin for oxygen, so more is released at the tissues. And another factor that affects affinity is temperature. So an increase in temperature causes a decrease in affinity because it increases the kinetic energy of the oxygen molecules, which causes them to dissociate, or they're more likely to be released. So in areas like the tissues where you're getting heat generated, if you've got a muscle that's exercising, you get heat generated, which causes a decrease in affinity and a release in oxygen. So it's worth just taking a look at the curve and considering taking some time and spending some time here to just kind of consider what the different factors cause. So a decrease in pH or an increase in H+ ions causes a right shift and a decreased affinity, and an increase in 2,3-diphosphoglycerate and an increase in temperature will also cause a right shift and a decrease in affinity. And then it goes the other way as well. So if you get a decrease in temperature or 2,3-diphosphoglycerate or a decrease in H+ ion concentration, you get an increase in affinity. Just takes a little while to kind of wrap your head around just which way each thing shifts. But the whole picture is that oxygen is released where you need it and taken up where it's like taken in the lungs. So carbon dioxide in the blood is transported in 3 forms. So it can be bound to hemoglobin, forming carbaminohemoglobin. It can be converted to bicarbonate ions, and it can be freely dissolved in the plasma. So much like how hemoglobin's affinity for oxygen can be affected by CO2, the carbon dioxide carrying capacity of the blood can be affected by oxygen concentration. So that's called the Haldane effect. So in this red line here, where you've got a higher partial pressures of oxygen, the total blood saturation of CO2 is actually lower. And this is because when there's high partial pressures of oxygen, it displaces the CO2 bound to hemoglobin and causes CO2 to be released. So that means in the lungs, where you've got high partial pressures of oxygen, it displaces the CO2 and causes it to be released to then get exhaled. But if you go to the tissues where there are low partial pressures of oxygen, where it's being used up, the CO2 carrying capacity of the blood is increased, which means more is taken into the blood to then be taken back to the lungs. So you've got these 2 effects here that kind of work together to make sure that the tissue is getting the oxygen that they need and that the carbon dioxide is getting taken away. So you've got the Bohr shift, which is the effect of CO2 and H+ concentration on hemoglobin's affinity for oxygen, which is this right shift here shown on this diagram. And then you've got the Haldane effect, which is when you have increasing partial pressures of oxygen, you have decreased CO2 concentration carrying capacity of the blood. And these 2 effects just work together to make sure that the body's getting what it needs and that you're getting as much oxygen taken in at the lungs and getting delivered to the tissues, and you're getting as much carbon dioxide carried away from the tissues into the lungs. So when you compare these 2 curves, you'll notice that the CO2 dissociation curve is linear, whereas the oxygen hemoglobin dissociation curve is a sigmoid curve. And you also notice that the oxygen hemoglobin dissociation curve has a plateau here, whereas the CO2 dissociation curve just continues as a straight line and doesn't really plateau. And that means that increasing ventilation will improve the CO2 exchange, but with an oxygen, there kind of reaches a point where increasing the partial pressure of oxygen doesn't actually increase oxygen saturation. And this means if a shunt develops, which was like in the previous picture, where you get an area of, where you get an alveolus that is under-perfused, increasing ventilation of the remaining alveoli can compensate for CO2 levels, but it can't compensate for oxygen levels, because increasing ventilation and increasing oxygen partial pressures, eventually you'll reach a point where you can't get more saturation of hemoglobin. So that's just the different way that the curves work. So I've got a practice question here, which is question 2, and it's which of the following best describes the Bohr effect? Sorry, it's quite a wordy question. I'll just give you guys time to read this through. And I'll start the polling. Okay, I'll just give you a couple more seconds there. Okay, so the correct answer is C, and that's the reduction in pH causes haemoglobin's affinity for oxygen to decrease and a right shift in the oxygen haemoglobin dissociation curve. Now, one thing to watch out for, which is, I'll just stop showing those results. One thing to watch out for is that an increase in H+ actually causes a decrease in pH. So just remember when you're talking about pH, it's a decrease that will cause a right shift in the curve and an increase in H+. So I've got a question in the poll here, a question in the chat, and that was, you said that with increased partial pressures of oxygen, you get decreased CO2 carrying capacity, but the line goes upwards. So I'll go back to that graph. So the graph here. So what this line going upwards is actually, so if you look at the red line, that's when your oxygen partial pressure is the highest, and that's 100. And the total blood CO2 is here, which would be lower. And the green line, which is where you have a higher partial pressure, the lower partial pressure of oxygen, sorry, you have a much higher total blood CO2. So when you're comparing the Haldane effect here, you're looking at the difference between the coloured lines. And the line actually going upwards here, if this is what you're talking about, is when the partial pressure of CO2 is increasing. So down along this axis here, the total blood concentration of CO2 is also increasing along. Does that answer the question? I hope. Okay, fabulous. So I will move on. Does anyone have any more questions about that section before we move on? No, that's fine. Okay, so now I'm going to talk about ventilation itself, and that's the actual movement of gases in and out of the lungs. So the process of inspiration and expiration requires movement of respiratory muscles. Now these include the diaphragm, which is the main muscle of inspiration here, of respiration. You've got your intercostal muscles, which are the muscles in between your ribs. And these have 3 layers, the external intercostals, the internal and the innermost intercostals. And you've also got accessory muscles of inspiration. And these are things like your sternocleidomastoid, your scalene, pectoralis muscles, serratus anterior, and your abdominal muscles as well. Now, I wanted this to be more of a physiology session, so I'm not going to go into the anatomy, but it's worth just in your own time kind of going over these individual muscles and working out when they contract and how that involves the movement of the rib cage. So the inner lining of the inner lining of the thoracic cavity and the outer layers, the outer edge of the lung, the space in between those two is called the pulmonary pleural space, and that's filled with pleural fluid. And that fluid creates surface tension between the lung and the inner thoracic cavity. And that means that the lung adheres to the inner lining of the thoracic cavity, which means when the thoracic cavity expands, the lung expands with it. And when it reduces in size, so does the lung. So in inspiration, what you get is you get the diaphragm contracting, and you get the external intercostal muscles contracting, and together that increases the volume of the thoracic cavity. And because of the surface tension, you also get an increase in lung volume. And this increase in lung volume causes a decrease of pressure inside the lung. And that's because you've got the same amount of air in a larger space. So you get a decrease in pressure. And once that pressure drops below the pressure of the external environment, you get air entering the lung down the pressure gradient. So you get gas drawn into your lungs during inspiration. And then during expiration, your diaphragm relaxes and your external intercostal muscles relax, and you get elastic recoil of the lungs, which causes them to decrease in size, and you get an increase in pressure inside the lung. And then once the pressure increases above the pressure of the external environment, you get air moving out of the lungs. So that's how you would gases exchange in a normal inhalation exhalation. But in situations where you want to get larger volumes of gas exchanged, for example, if you're exercising, you need your thoracic cavity to expand to a bigger size. And that's when you get your accessory muscle involvement. So in inspiration, not only would you have your diaphragm and your external intercostal muscles contracting, you also get these other muscles of inspiration here on this side of the diagram. They also contract to help increase the volume of the thoracic cavity even further, causing the lung volume to increase even further as well. And that just draws more gas into your lungs. And then during expiration, you would get relaxation of those muscles, but you'd also get the contraction of the internal intercostal muscles and the oblique muscles as well. And that just pushes the thoracic cavity volume, it like kind of squashes it even further and makes sure that the lungs are a smaller volume so more gas can be exhaled. So in normal, just normal quiet breathing, you just get the movement of the diaphragm and the external intercostals. But then when you have kind of forced expiration during exercise, you get contraction of the accessory muscles as well, just to make sure you get more gas exchange to supply the body with the oxygen and remove carbon dioxide. So you also have different lung volumes in the lung. And these are measured using spirometry. As you can see, this middle picture here is a spirogram. And they're just often, it's just that it involves a closed circuit where you breathe into a spirometer and it just measures the different lung volumes. So when I think about lung volumes, I think of them as 4 volumes that are stacked up on top of each other. So you've got tidal volume, and that's just the volume of air inhaled or exhaled in a normal quiet breath. So that's if you're not exercising, you're just sitting and breathing, you get tidal volume, and that's around 500 milliliters in the average person. And then you also have your inspiratory reserve volume, which is on top of your tidal volume. And that's the volume of gas that can be breathed in with maximum inspiration from the end of normal quiet inspiration. So the end would be about here. And then this is the extra volume of gas that you can inhale with your maximum inhalation. And you've got expiratory reserve volume, which is the other way. And that's just the volume of gas expelled with maximum effort at the end of normal quiet expiration. And your final lung volume is your residual volume. So with lung volumes, they're all discrete, they don't overlap, and they just stack on top of each other to form the separate volumes. But you also have lung capacities, and these are formed of volumes added together, and they overlap each other. So you've got your vital capacity, which is the maximum amount of air that can be exchanged in your lung. So that's the inspiratory reserve and the expiratory reserve volumes and the tidal volume added together. You also have your functional residual capacity, and that's the volume of air left in the lungs after a normal quiet expiration. So it's the expiratory reserve volume plus residual volume. And this functional residual capacity is important because it can be altered in different diseases. And your final capacity is your total lung capacity, and that's just the full volume of gas that can fit in the lungs, and it's all the volumes added together. So I've got another practice question here. So this is a 63-year-old male which suffers from emphysema, which causes the residual volume of air in his lungs to increase. Which of the following will therefore also be increased? So I'll start the polls on this one. So I'll just give you a couple more seconds on this. So well done, guys, there. Most of you got this one right. So you'd also get an increase in your functional residual capacity. So if I just go back to the previous slide, you can see that if he was getting an increase in residual volume, you'd also get an increase in functional residual capacity, because that's the residual volume plus the expiratory reserve volume. Okay. Now, does anyone have any questions on that section before I move on to the next one? And that's fine. Okay. So control of respiration. Control of respiration is mainly done by the respiratory centers that are found in the medulla and the pons. Now, different receptors around the body send information to these centers, and these include chemoreceptors, and these are formed of central chemoreceptors, which are found in the medulla, and they send information about the pH of the CSF, so the cerebral spinal fluid. And you also have peripheral chemoreceptors, and these are found in the aortic arch and the carotid sinus, and they send information about the pH of the blood and the oxygen partial pressure and the CO2 partial pressure of the blood. You also have pulmonary stretch receptors in the lungs, and these send information about lung expansion and how big and like how the lungs are changing in size. And you also have irritant receptors, and these are found in the lungs and in the trachea and bronchioles, and these kind of detect if there's any irritants there that might need to get coughed out. And you have things like muscle stretch receptors and joint proprioceptors, which are sending information to the respiratory centers about how the limbs are moving. So all these together set up like a little negative feedback loop. So you'd have a stimulus in your internal environment. For example, you might get a change in CO2 levels in the blood, and that would cause a change in pH. And this is detected by receptors like chemoreceptors. You also have lung receptors kind of telling the respiratory centers about what the lungs are doing, and also muscle receptors, for example, if you're exercising, that are kind of detecting that movement and sending that to the respiratory centers. So all those receptors are sending afferent information to the respiratory centers in the brainstem, and these send efferent impulses to the effectors, which would be your muscles of respiration. And this can cause them to increase or decrease alveolar ventilation in response to that stimulation, and then that can change, like can bring back the change to its normal level. So the pH of the blood is determined by H+ ion concentration level in the blood, which should be here, and therefore carbon dioxide levels of the blood also affect pH, because you can see that carbon dioxide reacts with water to produce H+ because it's acidic. So what you get is pH is determined by the carbon dioxide level in the blood. So if you get an increase in CO2 levels, you get a decrease in pH, and if you get an increase in CO2 levels, you get a decrease in CO2 levels, sorry, you get an increase in pH. Now, CO2 levels are directly proportional to the rate of production of CO2 over the alveolar ventilation rate. And basically what this means is that ventilation changes the pH, the concentration of CO2 in the blood. So if you have hyperventilation, you get a reduction in the CO2 levels in the blood, and that will cause a rise in pH. And if you get hypoventilation, you get an increase in CO2 levels in the blood, and therefore you get a fall in pH. And what this means is if you have, for example, if you have something that's inhibiting your respiratory reflex, so you have an opioid overdose, that depresses your respiratory reflex, and that can cause hypoventilation, and that causes CO2 to build up in your blood and it will also cause pH to decrease. But it also means that that the respiratory system can compensate for changes in pH. So if you have a decrease in pH of the blood for another reason, for example, in diabetic ketoacidosis, you get production of ketones, which are acidic, and that will cause a fall in pH. In response to this, you can have hyperventilation of the lungs, which will reduce CO2 levels in the blood and then cause a rise in pH to try and bring it back to the normal level. So the respiratory system can kind of act as a compensatory mechanism for changes in pH in the blood. So a practice question about that section. So this is a 32-year-old female who's having a panic attack. She's breathing very rapidly and feels dizzy and lightheaded. What would her ABG results show? So I'll just start the polling for this one. Okay, just finish that one there. Okay, great. So the majority of you guys got that one right. It's A, which is you get a reduction in partial pressure of carbon dioxide because you're breathing more rapidly, so you're blowing off more CO2, and that would cause a corresponding rise in pH because your blood is less acidic. Okay. So does anyone have any questions about any of the presentation so far? So I can go to the Q&A questions that have been asked at the start. I've got one here. Does gas exchange in the lungs depend on both concentration and pressure gradients? Sorry, I should make that more clear. So concentration and pressure gradient is the same thing. So when you're talking about gas exchange in the lungs, you're talking about partial pressures more than you do about concentration. So partial pressure is just how much of the gas that you have present in either the blood or either in the external air. So does that answer the question, hopefully? And then another one is, does a pneumothorax cause dead space in the lungs? If so, how? So a pneumothorax is when you have a collapse in the lung. You get collapsing of that alveoli, and I don't think you can even get air into that collapsed part of the lung. So you're not really causing dead space because there's no space in the lung for you to fill. So a pneumothorax, I don't think at that point you could even really talk about VQ ratios because you're not getting either ventilation in that part of the lung. So hopefully that answers those two questions. Does anyone else have any other questions they'd like to ask? Got one in the chat here. Someone said, missed the start. Oh, what do high and low VQ ratios? Okay, so VQ ratios basically is ventilation to perfusion ratio. So ventilation is how much air you're getting to the lungs, and perfusion is how much blood flow you're getting. And you want VQ ratios to be 1 because that means you've got the same amount of ventilation as you do perfusion. And a high and low ratio means basically you've got a mismatch between the two of those. And that just means you have less gas exchange taking place in the lung. Like this presentation will be available on Facebook if you want to go back and rewatch the start. So does anyone have any other questions? No, okay. That's fine. The last thing is I just have a feedback link at the end here. So if anyone would please be able to give feedback on this lecture, that would be a great help to me. So you can just scan this link or visit this website at the end here. And that's just the end of my presentation. So thank you very much for coming, guys. And all the best for your exams.